Within the article titled Timely and Spatially Regulated B and T Cell Repertoire Maturation During Human Fetal Development, published in Science Translational Medicine, Richavi and colleagues investigated how the immune system develops during human gestation. This was done by analyzing the differences between B and T lymphocytes during development between 12 to 26 weeks of gestational age. The results communicate a fascinating finding in the temporal and spatial regulation of B and T cells, and how this can be used to improve immune function for developing fetuses through various means, as this was a widely unresearched area. This research was performed based on a few assumptions, namely, timely and spatial regulation. The maturation of T and B cells is intricately controlled through temporal and spatial regulation. Temporal regulation ensures that specific immune components are expressed at precise developmental stages, while spatial regulation guarantees that these components are expressed in the correct anatomical locations. This coordinated regulation is vital for healthy immunocompetence. The T and B cell repertoire refers to the diverse range of T cell receptors, or TCRs, and B cell receptors, BCRs, that the immune system develops. These receptors are crucial for recognizing and responding to various pathogens, including viruses and bacteria. Immunocompetence is the ability of the immune system to mount a normal and effective response against infections. Achieving immunocompetence involves the successful maturation and diversification of T and B cells, which is a carefully regulated process throughout fetal development. The primary aim of the article is to provide comprehensive insights into the development of the adaptive immune system in the human fetus. But more specifically, also, one significant goal of the study is to characterize the diverse immune cell repertoires that emerge during fetal development. The article aims also to dissect the mechanisms behind the temporal and spatial regulation of B and T cell development, and also addresses gaps in current knowledge. The article strives to enhance our understanding of fetal immune development.
The first experiment in this study aimed to quantify immune markers at various stages of fetal development in order to understand how the immune system matures. To achieve this, the researchers used quantitative real-time PCR, QRT-PCR, in order to measure the number of specific immune markers present in the different fetal blood samples. The markers measured included SJTRECs, SJKRECs, and the rearrangement of IRSSKDEs. SJTRECs are circular DNA fragments found in T cells. Therefore, their presence correlates with the presence of mature T cells. SJKRECs are also circular DNA segments, but are generated in B cells during their maturation. Lastly, IRSSKDEs are sequences involved in the genetic recombination process of T and B cells during their development. QRT-PCR is a technique used in both diagnostics and gene expression measurement experiments. It follows the same thermal cycling steps as PCR, however includes an oligonucleotide probe or intercalating dye which measures the quantity of the gene being amplified through fluorescence measured at the end of each cycle. In this study, researchers used TACMAN DNA probes. These anneal to a section of the gene being amplified and consist of a reporter fluorophore, indicated as R in the diagram, and a quencher fluorophore, indicated as Q in the diagram. The quencher absorbs the wavelength emitted by the reporter until it is cleaved off by Taq polymerase during the extension step of PCR. Then once the reporter is free, it fluoresces, and this fluorescence is measured at the end of each amplification cycle. Plotting raw QRT-PCR data will provide a diagram such as the one on the slide, where the x-axis is the number of amplification cycles, and the y-axis is a measure of fluorescence. The cycle threshold value, or CT value, indicates the cycle at which the fluorescent signal exceeds a defined threshold, which is unique to each experiment. A lower CT value signifies a higher amount of the target DNA, as less amplification cycles were needed to reach this threshold quantity. To accurately estimate the number of gene copies from fluorescence measurements, the researchers in the study constructed a standard curve using serial dilutions of the different immune marker plasmids, ranging from 10 to the 3 to 10 to the 6 copies. By comparing the CT values of the patients to the standard curve, they could determine the number of gene copies present. And to assist them in this process, they used a quantification algorithm. The primary limitation of QRT-PCR is that detecting very low levels of gene expression is challenging, as it requires a very high number of cycles to reach a CT value where fluorescence can be accurately measured. Additionally, the cost of this technique is significant due to the high expense of purchasing DNA probes specific to the gene of interest in the study, as well as the specialized equipment, which is also quite pricey. Finally, in addition to the limitations of QRT PCR, this technique was performed in duplicate, which can affect the accuracy and the reliability of the results.
The key findings from this study demonstrate a progressive increase in both TREC and KREC levels with increasing gestational age. Looking at graph A and graph B on the left, we see the expression levels of TREC and KREC genes over gestational age from 12 to 26 weeks. Both graphs follow a similar curve, the main difference being that the KREC levels appear before the TREC levels do, which indicate that B-cell lymphoesis occurs prior to T-cell lymphoesis during fetal development.
Contribution to society. So this study opens a gateway to a better understanding of the fetal immune system and at what stages it develops. In Australia, in 2021, 3.4% of perinatal deaths and 4.2% of neonatal deaths were due to infection. So gaining this better understanding of fetal immune systems opened doors for research in limiting these fetal deaths by providing targeted immune support. Vaccination strategies. So, by knowing and understanding when the active immune system develops, scientists and doctors can make informed decisions on infant vaccination strategies and optimize vaccination plans based on the stages of development and understanding how elaborate the infant's immune repertoire is at any point in time. Immune deficiency. So, autoimmune diseases affect roughly 5% of Australia and New Zealand's population. So, by having a working understanding of the origins and development of the T and B cells, researchers can establish the origins of immune deficiency disorders and develop strategies for early diagnosis and prevention. And education and awareness. So, above all, this study informs both public and professional understanding of the developing immune system. And regardless of the effectiveness of the study in future continuations, it's a massive stepping stone in our understanding and research, as well as the method and techniques being able to form future studies. Limitations. So, the main limitation of this study is sample size. The study has limited access to healthy gestational fetus blood samples. So, due to limited fetal DNA, parts of the experiment had to be completed in duplicate rather than triplicate, leaving a larger room for error in identifying trends and patterns. The sample size was dictated by the rate of sample selection over a year, and any samples collected from a fetus known to interfere with immune development or relevant chromosomal abnormality were excluded, leaving an even smaller number of samples. 5 fetuses did have genetic abnormalities, however, these were determined to not affect the immune system, so were left in the study. Future directions, further understanding of the mechanisms of development. So now we have an understanding of the temporal and spatial aspect of T and B cell development. We know when and where development takes place, so now we can study how it takes place, such as the transcription factors and signaling pathways involved. So this research was only possible due to technological advancement between previous studies. And as technology continues to develop in the future, we can gain more specific knowledge about the development of the active immune system. Epigenetic regulation of T and B cells. So epigenetic regulation is the mechanism that activates and deactivates cellular processes. Epigenetic dysregulation of T and B cells contributes to and is likely the cause of the development in a number of autoimmune diseases, such as systematic lupus erythematosus, or SLE, which you may have heard of from Selena Gomez. So, abnormal epigenetic regulations in B cells majorly contribute to the pathogenesis of SLE. So, by knowing the temporal and spatial development, so the exact time and place the events of the immune system develop, further research can be done on epigenetic regulation and at what moments these mechanisms are going wrong. And this will have applications into the understanding, diagnosis, and treatment of autoimmune diseases, as well as applications into cancer treatment and research. Longitudinal studies. So, by conducting another study over an extended period of time, it will be possible to follow fetuses from before birth into childhood to understand how the early immune system will impact long-term health outcomes. So, overall, this study aimed to quantify immune markers at various stages of fetal development and gain an understanding of the timely and spatially regulated maturation of T and B cells. Through use of quantitative real-time PCR, the study was able to discover that B cell development precedes T cell development and formulate the process of fetal immune development through increase of receptors with gestational age. Despite limited sample size, this study was able to fill gaps in our knowledge and understanding of fetal immune system development and offer future direction in further research of mechanisms of development, as well as the epigenetic regulation of B and T cells. This study has majorly contributed to society through public and professional understanding of immune development and a knowledge for informing vaccination strategies.